Hi there, my name's Joshua and I'm a neuroscientist and biotechnology student at the Swinburne University of Technology, and today I will be talking to you about both my STEM idea and a research project that I'm working on: stem cells to glial cells, advanced disease modeling and therapeutics. So, what is a glial cell, I hear you ask? Well, glial cells, or neuroglia, are non-neuronal cells found in both the peripheral and central nervous systems. Unlike neurons, they do not produce electrical impulses. Rather, they are a set of support cells that protect and supplement neuronal functions. As an analogy, if neurons are the Beyoncé of the brain, then the glia are her entourage. Sure, Beyoncé may be fabulous as she already is, but without the support of background singers, lighting and effects teams, and makeup artists, just to name a few, the final performance will be somewhat lackluster. Did you know that post-mortem histological analyses showed that Einstein's brain, which, due to his short stature, was smaller than average, had a higher glial to neuron ratio than the average male brain? So, this provides some evidence, albeit limited, that glial cells are important for intelligence. In the central nervous system, the following glial cells are found: oligodendrocytes, which produce the myelin sheath to insulate the axon; ependymal cells, which form the neuroepithelium of the brain's ventricular system, producing cerebrospinal fluid; microglia, which are the resident macrophage cells and serve as the brain's immune system; and astrocytes, the focal cell in this study, which, among many other functions, support the blood-brain barrier. In this project, I will attempt to demonstrate that human pluripotent stem cells, a type of master cell, can be differentiated into astrocytes through gene cloning overexpression of two transcription factors, Sox-9 and NFIB. In essence, there are two main aims of my research project. One, to successfully clone transcription factors from one vector to another, which uses gene cloning technologies. And two, to use these artificial vectors to program stem cells to differentiate into astrocytes. The impetus for this study was to generate human astrocytes that can be used for disease modeling, namely Alexander disease, a leukodystrophy disorder caused by glial fibrillary acidic protein, GFAP, gene mutations, which is behaviorally characterized by intellectual disability and spasticity. In Alexander disease, this compromised astrocytic function causes destruction of the myelin sheath that surrounds and insulates axons. It is histologically characterized by abnormal rosenthal fiber protein deposits in astroglia. A 2018 study used human-induced pluripotent stem cells derived from an Alexander disease patient to show that GFAP mutations in astrocytes leads to impaired oligodendrocyte proliferation, the fundamental myelinating glial cell in the central nervous system. However, the methodologies used in my research project are not without their limitations and controversies. Stem cell research, gene cloning, and animal studies are hot-button topics in science and ethics. For example, one potential limitation is that non-human animal models of AXD, such as rodent models, do not sufficiently recapitulate dysfunctional myelination in human patients. Therefore, therapeutic interventions following in vitro disease modeling is a potential use for human and not non-human animal stem cell proliferation technologies. Moreover, human embryonic stem cells are not without their controversies. Utilization of embryonic stem cells, the most common subtype of pluripotent stem cells, relies on the destruction of the pre-implantation stage embryo during fetal development. Fortunately, my project will use rodent stem cells, but nevertheless, the problem of rodent to human extrapolation still exists. The future of stem cell, gene cloning, and neuromedicine seems rather bright since undertaking this research project. Disease modeling using artificially derived pathological human cells may allow us to study human diseases in vitro without relying on animal experimentation or post-mortem analyses. For next time, I'm Joshua Nichols, and thanks for listening to my STEM idea.